Thank you very much, the President of NISS, the Registrar, our distinguished Vice Chancellor, soil scientists, distinguished participants, soil scientists. I'm delighted to present this lecture, being the third, and I believe it will make a lot of meaning to us in the Institute, and of course, in the development of agriculture in Nigeria. Okay, thank you very much, the Registrar. Once again, I welcome everybody to this lecture, or to this training in a way. And the top, what is soil fertility evaluation? It is to know the nutrient supplying power of native and residual element nutrients in the soil which are available to the crops in a particular soil, and the need or otherwise to add additional nutrients. And therefore the advantages can be seen. Advantages in evaluating the soil to know its fertility status can be seen as an aid in maintaining and improving soil productivity. It is a key for adequate and balanced fertilization in crop production. And balanced nutrients supply to balanced fertilization refers to the application of essential nutrients in the right amounts and proportions using correct methods of assessment and using correct time of application suited for specific soil crop climatic conditions. In other words, in soil fertility evaluation, we have to aim at balanced nutrition for the crop. We have to aim at balanced fertilization so that whatsoever we apply from external will not overwhelm the crop, will not overwhelm the soil. Next slide, please. Next slide. What are the techniques? 4 techniques are listed here: soil testing, plant tissue analysis, biological test, and nutrient deficiency symptoms. Sometimes we talk about soil testing. We also talk about soil analysis. You can analyze soil for the purpose of just knowing its characteristics: physical analysis, chemical analysis, and biological analysis. When we're talking about soil testing, we aim at stating the soil need for additional nutrients for the purpose of growing crops profitably. So soil testing is a chemical analysis that provides a guide, that provides guideline for addition of amendments or fertilizer to the soil. The primary advantage of soil testing is determining the nutrient status of the soil before the crop is planted, as compared to plant analysis. The objectives are many. We can do soil testing for the purpose of classifying soil into different fertility groups, for preparing soil fertility maps of a given area, and sometimes recommending or suggesting fertilizers practices to be applied. It is also important for the prediction of likely profitable crop response to applied nutrients. It is also important for the assessment of the type and degree of soil-related problems like salinity, solidity, acidity, and suggesting appropriate reclamation strategies. What are the steps involved in doing this? We sample for soil analysis. The first method of soil fertility evaluation is soil testing, which involves analysis. It requires collecting sample. It requires preparing soil sample. It requires analytical procedure. You extract and determine or measure. Sometimes we do calibration, and sometimes we also do calibration and correlation, and we interpret the results before we make fertilizer recommendations. So for this lecture, we shall look at a few of these steps involved. Sampling. Soil sampling is perhaps the most vital step for any analysis.
Yes, if you click on it, you see that
because a very small fraction of the huge soil mass of a field is used for analysis and converted on the basis of hectare. So it becomes extremely important to get truly representative soil samples from the field. We normally prepare soil sample by drying, grinding, sieving, according to the needs of the analytical procedure. For instance, we are going to determine nitrogen, total nitrogen, and other forms of nitrogen, you have to grind. Analytical procedure include using suitable method in which satisfies the following 3 criteria. It should be fairly rapid so that the test results can be obtained in a reasonably short period. The analytical procedure should give accurate and reproducible results of given samples with least interference during estimation. It should have high predictability, that is significant relationship of test values with crop performance. We talk about correlation. We talk about correlation, and we talk about calibration. In doing soil tests, selected nutrients are analyzed using chemical methods differently. Total nitrogen, we normally use Kjeldahl method. It has its own merits. And the merits, the method is time-consuming, lengthy, and costly. The rate of mineralization of nitrogen varies with the soil. So there are, you use macro Kjeldahl units, you use macro Kjeldahl units. There is a flow diagram as part of this presentation, which shall be displayed later on. Organic carbon enables us to determine organic matter, which is the storehouse of soil fertility, and we normally use Walkley and Black method. We also do loss in ignition. Available nitrogen by the alkaline potassium permanganate extraction method. Next slide. Next slide, please. Available P2O5, available phosphorus, Olsen method for alkaline soils and Bray method for acid soils. In Nigeria, we are using Bray 1 and Bray 2. If there will be issues, we shall raise accordingly. The Bray method that we use in Nigeria is for acid soils. It has high efficiency using acid fluoride in dissolving P, useful in acidic or slightly calcareous soils. We also do available potassium using ammonium acetate extraction. The merits and demerits are there. Next slide. Available sulfur using calcium chloride, and sometimes we use heat-soluble sulfur. The heat-soluble sulfur extracts water-soluble and organic sulfur, but it's time-consuming and lengthy. I don't know how many laboratories in Nigeria do this, but it's one of the methods that is used in determining sulfur availability in soil testing. Available macronutrients, we use DTPA, extractable, and sometimes we use dilute HCl. Next slide. After we have done extraction and an assessment of the nutrients in the soil, we sometimes do calibration. Calibration is required for interpretation of results. And calibration is a means of relating what is in the soil test, relating the soil test value and yield response from added nutrient to the soil. To calibrate, you have to look at establishing a relationship between a given soil test value and the yield response from added nutrient to the soil. That means you have to plant in the greenhouse, in the field, and you calibrate. And calibration is needed for you to be able to recommend nutrients. So you try to look at different nutrient levels, and you now look at the optimum yield, or what you can call, well, maximum yield. And to do this, you plot soil test values against the percentage yield to calculate the relationship with the equation that is given here. Crop yield with adequate nutrient minus crop yield without nutrients, that is the control, over crop yield by adequate nutrients, and you multiply by 100. Now, there's also critical level of nutrients in soils. This is a categorization of nutrients so that you can take decisions easily. And this has become very useful now with the soil test kits that NISS is trying to make it, penetrate Nigeria. And I can tell you that farmers and ADPs are very excited about them. So if after extracting the soils with these extractants, soils are categorized on the basis of low, medium, and high content of individual nutrient elements, and decisions can be taken for these nutrient elements: molybdenum, boron, copper, zinc, manganese, iron, sulfur, potassium, phosphorus, and nitrogen. This classification enables decision to be taken during actual fertilizer application. When you have soils falling within low range, low classification of soil nutrients, it is recommended that you add additional fertilizer, additional 25% more than what is recommended. If you have medium range, you may not need to add because the response may not be there. If you have high range, you need to reduce the fertilizer addition to make economic sense. Next slide, please. We now move to plant testing. Generally, we have talked about soil testing, soil analysis for the purpose of recommending fertilizers. We also use plant testing. There are many ways of doing this. Analysis of tissues from plant growing on the soil. Plant tissue analysis is the determination of the concentration of an element in the plant sample from a particular portion of the crop at a certain time or stage of the morphological development. So you sample plants when it is growing, and you do analysis. The plant analysis has been used as a diagnostic tool or complementary tool to soil testing because in many situations, the total or even the available content of an element in a soil fails to correlate with the plant tissue concentration or the growth and yield of crop due to many reasons, including physicochemical properties of the soils and the root growth patterns. So while you do soil analysis and you want to do that alone, you can complement that with plant testing procedures. On the other hand, the concentration of an element in the plant tissue is positively correlated with the plant health. Therefore, plant analysis has been used as a diagnostic tool to determine the nutritional cause of plant disorders, stroke diseases. So when once in a while you see some plant behavior, you can do plant tests to get a relationship, to establish to take some decisions. Earlier on, I had mentioned correlation as one of the things we do to take decisions. And of course, you need to plant the crop, a test crop in pots in the lab or in greenhouse, or in greenhouses, or in the field. And you have to analyze the source, the plant sample, to know, to start and do a correlation to establish relationship between the amount of nutrient extracted from the soil by lab tests and nutrient uptake by plants under greenhouse or field conditions. So you must do plant tests as part of the overall soil test crop responses relationship, soil test crop responses activities. So the aims of plant analysis to testing include diagnosing or confirming confirming the diagnosis of visible symptoms. Some symptoms are caused by nutrient problems. Some symptoms are caused by biological factors. Some symptoms are caused by biological factors that are not necessarily nutrients. So plant analysis can enable us to diagnose this. Identifying hidden hunger in plants. There can be nutrient in the soil, but the plants suffer from hunger. It's called hidden hunger. Plant may just have some physiological challenge, and it cannot take up the nutrients. So you don't need to continue to add nutrients, except you're able to identify the problem that may be physiological. Indicating whether applied nutrients have been taken up by the plant. Again, we have said it earlier on that you need to do plant analysis to know what volume of nutrients the plant has taken in relation to what is in the soil. Indicating interactions or antagonisms among nutrient elements. Nutrients can interact to enable a nutrient or a set of nutrients to be useful. Nutrients can also antagonize one another. So you need plant analysis to know if there is such situation. You have diagnosis of nutrient deficiencies, toxicities, and imbalances. Deficiencies can be diagnosed through plant analysis. Toxicities can be diagnosed, and imbalances too. Measurement of the quantity of nutrients removed by crops to replace them in order to maintain soil fertility. You can use plant analysis to know the rate of removal of nutrients in your crop, in your field. Estimating overall nutritional status of the region or soil types. So you can use plant analysis to make an estimate of the fertility status of regions or zones in a place, in a state, in a local government, in a village, even in a school. What are the steps in collecting? What are the steps in doing plant analysis? As usual, you have to collect the representative samples from plant parts at different stages. Sometimes you need to wash, dry, and grind. You can do oxidation of the ground plant samples to solubilize the elements. And you do estimation of different elements by the process of chemical extraction and analysis. And of course, interpretation of the status of nutrients with respect to deficiency, sufficiency, toxicity on the basis of known critical concentrations. There have been established critical concentrations in plants. There have also been established critical nutrient levels in the soil. Collection and preparation of plant samples. Next slide, please. Next slide. Next slide. Next slide. We are required to collect representative plant parts at a specific growth stage because nutrient concentration in plant vary with growth stage, and it reflects the nutrient concentrations at particular growth stages. You need to collect the recently matured, fully expanded leaves just before the onset of the reproductive stage and put in perforated paper bags, not in airtight containers because of respiration and vaporization and heat. The plant samples are often contaminated with dust, death, and residues of the sprays. So it needs to be washed first under a running tap water, followed by rinsing with dilute HCl, very dilute distilled water, and finally in the ionized water. These are all steps in ensuring cleanliness of the samples, in ensuring quality control and quality assurance, and ensuring reproducibility of results. The wash samples are dried in a hot oven to temperature of 60 degrees, plus or minus 5, for a period of 48 hours. And ground in a stainless steel mill to pass through a sieve. Next slide, please. Part of what we do is oxidation of plant material. The main objective of oxidation is to destroy the organic components in the plant material to release the elements from their combinations to inorganic forms. You can do this by dry ashing at the controlled temperature in muffled furnace. You can do it by wet oxidation using an acid or a mixture of acids. Next slide. Next slide. Dry ashing. The dry ashing involves putting the plant material so ground in silica crucibles and ashed in muffled furnace. You just char it, it becomes powdery ash. 3 to 4 hours. The temperature is an important consideration in dry ashing because nitrogen and sulfur, being highly volatile, is lost during ashing at that. So ashing is not suitable for many elements. The ash is dissolved in 2 ml of conch, in 2 ml of 6 normal HCl, heated on a hot plate to near dryness and again dissolved. These are all procedures. The extracts may contain different amounts of insoluble materials, mainly silica, depending on the plant species. If you are doing grass plants, sugarcane, forage grass, you are likely going to have a lot of silica in them. And then this becomes part of what you consider in the analysis. Keep these extracts for some time to settle down, to settle insoluble materials, and separate by filtration before extimation. You can use centrifuge to do all method from these extracts by suitable techniques. The result obtained by this method are quite satisfactory and comparable to those obtained by wet digestion processes. Moreover, boron can only be determined by dry ashing since it's volatilized during wet digestion with di and triacid mixtures. Next slide. When we now move to wet digestion, this table shows us the various acid combinations and where it is most applicable, with some remarks. We are going to use concentrated sulfuric acid and nitric acid. You can wet oxidize or wet digest any vegetable material. The same thing with sulfuric acid and hydrogen peroxide. You can do any vegetable material. Nitric acid, anything of biological origin, including animal tissues. If you are using perchloric acid and sulfuric acid, anything of biological origin. Nitric acid, protein and carbohydrates. And if you are going to use 3-acid mixture, it has universal applicability. And the remarks are there. Acid double acid, sulfuric acid and nitric acid are the most common, commonly used in over here. Next slide, please. Generally, nitric acid, perchloric acid, and sulfuric acid are used in weight digestion method. This acid are used either singly or in combinations of 2 or 3 in ratios that are given here. So it all depends on what individual laboratories have been doing. And we shall, with time, be able to do harmonization of methods in Nigeria through the instrumentality of Nigerian Institute of Soil Science. The powdered plant samples are dissolved in these suitable diacids or triacid mixtures using hot plates at very high temperatures. We normally cool the content and add some distilled water. We filter and make desired volume. The filtration can be used for estimation of different elements. We have prepared a flow diagram for this. Okay, this is the flow diagram. Let's see if you can see the flow diagram.
1 hour.
Next slide. The flow diagram is not very visible, but what is here is showing a soil sample on the left side and plant sample on the right side. All the processes of oven drying, sieving, extraction, leaching, and testing for both plant and soil samples are shown here. But the base of the flow diagram show the nutrients and possibly the method of determining titration using Kjeldahl, using ion selective electrode, using flame photometer, using spectrophotometer, using atomic absorption spectrophotometer for all the nutrients listed here: lead, nickel, cadmium, zinc, manganese, copper, iron, molybdenum, copper, cobalt, nitrogen, potassium, magnesium, calcium, sodium, potassium, nitrogen. Nitrogen, you can determine total nitrogen and nitrate forms of nitrogen through Kjeldahl method and ion selective electrode. Next slide, please. This slide shows Kjeldahl analytical steps. It shows Kjeldahl analytical steps. It's a bit tiny, but all the steps, when you get the hard copy or soft copy, you can look at it again. It's just showing the guide in how to do Kjeldahl analysis. There are very many things to do at the end. You have automatic metering of solutions. You have an automatic way of doing things, and several things are done automatically at the base. Next slide. As still part of soil plant testing. Next slide. As part of plant testing, tissue tests are rapid and essentially qualitative. The nutrients are absorbed by roots and transported to those plant parts where they are needed. The concentration of cell sap is usually good indication of how well the plant is supplied at the time of testing. The plant parts, usually leaves are removed and plant sap is extracted. The plant sap is usually tested for nitrate, phosphorus, potassium by colorimetric method. Now you can do rapid tissue test. You can collect plant sap and do a rapid test through colorimeter using colorimeters, and you can take quantitative decisions by looking at colors and nutrient grading and fertilizer recommendations. Next slide. There is a not too popular biological approach called DRIS, Diagnostic Recommendation Integrated System. It's not very popular in Nigeria yet. In this approach, generally plant samples are collected from farmers' fields. These plant samples are analyzed for nutrient content and they are expressed as ratios of nutrients with others. The nutrients whose ratios are not optimum for high yields are supplemented by top dressing. This approach is generally suitable for long duration crops, but it is being tested for short duration crops like soybean and wheat. It's popular in the East, Bangladesh, India, I think in the US. We have other biological tests. In biological tests, the growth of higher plants or microorganisms are used as a measure of soil fertility. The biological methods consist of raising a crop or a microbial culture in a field or in a soil sample and estimating its fertility from the volume, that is yield or mass of the crop or microbial count. So certain crops can be planted in cultures or microorganisms can be planted, and the microbiologists will be in a position to do microbial count and be able to see the yield relationship and make interpretations and recommendations. These methods can be used for direct estimates of soil fertility. These methods are time-consuming, therefore not well adapted to the practice of soil testing in this part of the world. As part of biological tests, we commonly do field tests. The field plot techniques essentially measure the crop response to nutrients. NISS and the OCP have done that last year and this year, where certain recommendations, nutrient source to give particular nutrients are experimented and compared using cassava or maize or rice. In this test, specific treatments are selected and randomly assigned to an area of land which is representative of the conditions. Replications are used to obtain more reliable results and to account for soil fertility. Field experiments are essential in establishing the equation used to provide fertilizer recommendation, optimum profitability, and impact of nutrient use on the environment. So field experiments, field tests using crops and select nutrients or select recommendations or select rates of nutrient source or select rate of nutrients is very useful, and we have been doing that as a matter of routine. We also do pod cultures in biological tests. The pod culture test utilizes small quantities of soil to quantify the nutrient supplying power of a soil. Selected treatments are applied to the soils and the crop is planted and evaluated. We normally use maize. Maize is very sensitive to this kind of thing. Crop response to the treatments can then be determined by measuring total plant yield and nutrient content. Sometimes we took the plant part, the overall mass, or sometimes we wait until the plant yields flowers or fruits or seeds. A lot of time, we can just take the whole plant, including the stems. Next slide. We also do laboratory tests as part of biological tests. This nubile seedling technique is based on the uptake of nutrient by growing a large number of plants on a small amount of soil. I have not done this before, but I spotted it in the text and I felt it should be mentioned here. The seedlings exhaust the available nutrient supply within a short time. The total nutrient removed are quantified and tables are established to give the minimum value of nutrients available for satisfactory yields of crops. So what you put, the plant takes, and you look at what is remaining in the soil, and you make inferences and make recommendations. Next slide, please. Microbial method. Next slide. In the absence of nutrients, certain microorganisms exhibit behavior similar to that of higher plants. Azotobacter or aspergillus have been known to be useful in reflecting nutrient deficiency in the soil. The soil is rated from very deficient to not deficient in the respective elements, depending on the amount of colony growth. So you grow azotobacter, you grow aspergillus, depending, and you look at their growth pattern, you look at the bloom, you look at their overall mass and colonies, and you count and you compare. In comparison with other methods that utilize higher plants, microbiological methods are rapid, simple, and require little space in the laboratory, in your house. If you have a small lab, you can get a small lab in your house, you can do it. These laboratory tests are not commonly used here. But it's commonly used in many other places in the world. The fourth aspect of soil fertility evaluation is observing symptoms on plants. And we observe symptoms and try to relate those symptoms to nutrient deficiency, are different from symptoms that are biological or symptoms that arise from insects or from any other source. So what are the issues in nutrient deficiency symptoms? The plant requires 17 essential nutrients for their optimum growth and development. And then one of the 17 is nickel. Nickel is not properly studied, but it has been identified and added as the 16th after chloride. Hydrogen, calcium, and oxygen are supplied from the air. Nitrogen has a way of coming from the air to the soil, and plants take them more from the soil. So most of the essential nutrients that are supplied by the soil express symptoms which trained personnel, experienced persons, and people that are directly involved in observing plant nutrient deficiency symptoms have used effectively and efficiently. It is a good tool to detect deficiencies of nutrients in the field. When the plant nutrient is below critical concentration in the plant, it shows deficiency symptoms. These symptoms are nutrient specific and show different patterns in crops. You need to be very well trained to do this. It has limitations though. The visual symptoms may be caused by more than one nutrient. Deficiency symptoms may be due to an excess quantity of another. Deficiency symptoms in the field may be due to disease or insect damage, which can produce certain micronutrient deficiencies. Nutrient deficiency symptoms are observed only after the crop has already suffered an irreversible loss. Sometimes you can correct, particularly when it comes to nitrogen deficiency at the early stage. And two, if you are not color sharp, you must also have challenge as a person. Look at nutrient deficiency symptoms. Plant observation and visual diagnosis. We need to check the extent of deviation from the full color. A normal dark green color symbolizes sufficiency of nutrient supply, while light yellowish tinges indicate deficiency, so long as other factors such as diseases, insects are not responsible. Check deficiency symptoms against color photographs of known symptoms in a given crop. Use growth differences between fertilized and unfertilized crops. we have a checklist of deficiency symptoms. Symptoms that appear first on older leaves: chlorosis, starting from leaf tips, you suspect nitrogen; necrosis has dead spots on leaf margins, you suspect potassium; chlorosis mainly between veins, magnesium; brownish to grayish whitish spots, SPOT, please. You suspect manganese, particularly in cereals; and reddish color or green leaves or stem, phosphorus. Symptoms appearing first on younger leaves: mottled yellow-green leaves with yellowish veins, sulfur; mottled yellow-green leaves with green veins, iron; brownish black spots, manganese; youngest leaf has white tip, copper; youngest leaf is brownish or dead, in cocoa, boron. Now we can see if we can make any sense of these photographs by Epson and Bloom, 2004. There was an experiment to see deficiency symptoms attributed to specific nutrients. Nitrogen. Here there is an attempt for uniform yellowing. Chlorosis, mature leaf show pale yellow color. If the leaves are mature, the yellowing is pale yellow. If the leaves are young, you see bright yellow coloration and uniformly on the leaves. For phosphorus, you have some necrotic spots with strong purple tints. Plant becomes stunted or dwarf. Phosphorus has the tendency to hasten crop development. So if it is in excess, expect crops to mature very early. If it is deficient, crops become stunted and does not grow to its peak. Next slide, we have potassium. In potassium, you have marginal necrosis and tip browning. You have short internodes. And in cereals, you have elongated blades. These are symptoms of potassium deficiency. But potassium is one nutrient that does not express problem in excess. So you can take as much as is available. And we normally call it luxury consumption. Calcium. On that diagram, you see leaf necrosis around the base of the leaf. Wilting of leaves is a common feature in very severe cases. You will see the leaf wilting, and you have leaf browning and leaf dead leaves at the base. The next slide is a picture showing magnesium deficiency, leaf interveinal chlorosis. It is all uniform chlorosis. The chlorosis are around the veins. And you also have necrosis. You may have red and purple tints, along the leaf blade. Next one is sulfur. Sulfur distinct reddish color of leaves and petiole. You're going to see some coloration in the case of sulfur deficiency up to the petiole. But overall yellowing is an evidence of sulfur deficiency with specific coloration reddish up to the petiole. Zinc. Zinc shows an advanced stage of interveinal necrosis, dead portions, very, very conspicuous. And of course, they can be colored. Molybdenum. Molybdenum shows mottled spots along with interveinal chlorosis, yellow along the veins, but you have mottles. Mottles are if you have to splash mud on the wall, there is no particular pattern. It's all scattered like that. Can have little fingers of patches. Iron. Iron shows strong chlorosis at the base of the leaf with some green netting. You have patches at the base of the leaf. You have chlorosis, not uniform chlorosis like nitrogen deficiency. So you have strong chlorosis at the base of leaves, and you have also green netting. In other words, you have the veins are seen at the background. Manganese. Manganese show light intervernal chlorosis on young leaves, similar to iron deficiency. With mature leaves, mottled, again, you have wilting and curling. Copper expresses its symptoms more by curling. Curl leaves with petiole bending downwards, wilting. The petiole will droop as if there is a physiological drought. So it is because copper distorts water movement, and you have the leaf loses its turgidity. It becomes less. The toggle loss, it loses its toggle, and the young leaves, appear to be drooping. Boron. Boron, generally yellowing, but it's light. And young twigs. It affects both the leaves and the twigs. And finally, chloride. Abnormal shapes of leaf with distinct intervernal chlorosis. You can see lack of chlorine there. Chloride ion in the plant medium, and plants show abnormal leaf shape, and intervernal chlorosis very, very distinct. Sometimes we have nutrient toxicities. When nitrogen is so much in the growth medium, plants develop very deep green color, and it's up to the twigs. The vegetative buds are more developed than the reproductive buds. And ammonium as a nutrient, as ion, as the form of nitrogen uptake, causes root to turn brown with necrotic tips. So there can be ammonium toxicity affecting the roots. When the root tips are affected, root hairs are affected, uptake of nutrients and water becomes a problem. When nitrogen is too much in the plant system, the plant spends more time developing the vegetative portion at the expense of the reproductive part. And in some cases, it is prone to diseases and lodging. Phosphorus excess can cause iron and zinc deficiency. We talked about nutrient antagonism last time. When you have excess phosphorus, it distorts the availability of iron and zinc. Potassium, yes, even though I said earlier on that potassium doesn't cause too much problem, but it has been shown to cause calcium, magnesium deficiencies in some very rare cases. Magnesium, when you have magnesium in excess, very high magnesium can cause calcium deficiency. The problem starts from the lower leaves and develops to the main stem. Calcium, very high calcium can cause magnesium and boron deficiencies. It is still talking about nutrient antagonism. Manganese also causes reduced growth rate, necrosis along membranes, leaf die back to the stem. Leaves start to die until they get to the stem and they drop. The more the leaf drop, the lower the photosynthesis activities and the lower the yield. Boron. Toxicity symptoms show as yellow tinted band around the leaf margins. The chlorotic zones become necrotic and gray. The major parts of the leaf remain green. So these are all issues associated with toxicity after we have seen deficiency. Ladies and gentlemen, I think I can stop here as I thank you very much. Thank you. Thank you.
Thank you very much, Professor Ibia. Is Professor Raji back? Hello? Is Professor Raji back? Hello? Hello? Is Professor Raji back? Hello?
I'm not sure it's on the platform, sir.
I see. Well, I believe, can you give what is on the agenda? Are we not to ask some questions?
Yes, sir. It should be question and answer session.
And comments.
And comments.
Yes. Professor Waka's hand is up.
Professor Waka,
yes, sir. My hand is up.
Registrar, can you take up this for us? Mr. Registrar? Nobody is answering me now. Okay, Professor Waka, please go on.
All right, thank you very much, sir. First of all, I want to appreciate the lecturer for a good work. But my emphasis is on soil sampling. My emphasis is on soil sampling. One of the irregularities we have noticed in our publications is lack of knowledge of the soil. Now, for soil sampling, you see that people are using different methods. We need to have a good knowledge of the soil. Now, for somebody to go to the field to sample, we will have to know what type of soil.
Please, if you are not talking, please mute yourself.
Okay. Do I continue?
Some worker, please continue.
Okay, that's right.
Yes, please.
So there's a need to know the soil. People just go to the field for soil fertility. Are they taking profile samples? Are they taking composite samples? If they are taking composite samples, how many what area will a composite sample cover? All these things are not made clear. So the soil fertility division to please try to make it clear to soil scientists what the steps to take.
Because others, mute yourself. Mute yourself. Yes, have you finished? Professor Nwaka, have you finished? I can see Professor Ashadu's hand on. Can the lecturer be please writing down this so that we will not be taking time to answer one by one?
Thank you very much, Mr. President. First of all, I think some people are not obeying instructions. To move themselves, they will not move. Some people have a lot of distraction behind. So we should learn how to obey instructions. Well, I thank Professor Ibiam for well-detailed lecture. He has given us lots of information, which, anyway, for those who are not in the real area of soil fertility, a lot of information. For soil sampling, like Professor Mwaka pointed out, I think he talked about representative soil sampling, because once one knows the land configuration in terms of slope, areas, and that of different vegetation and so on. Since it's for soil fertility evaluation, I don't think whether going for soil profile digging and so on would be necessary now for soil fertility evaluation. But where we sometimes differ is depth of sampling. For instance, some people talk about 0 to 15 and 15 to 30. But generally, I think the normal thing, or what I think the majority do now, is 0 to 20 and 20 to 40, in order to accommodate reasonable number of arable crops. So I think he has done a lot, and I want to commend him for the time taken to put down all those things. Mr. Chairman, thank you very much.
Thank you.
Yes.
There's one Garba Omar. Who is Garba Omar? Your hand is up. You want to say something? Garba Omar?
Yes, can you hear me, sir?
Yes.
Okay, can I go ahead?
Please go ahead.
Okay, I just want to share one very important information.
Chairman, sir, Professor Jamalo is on the line.
The deficiency symptom of nitrogen and sulfur in the fields are really very similar. So for you to be able to distinguish whether the field is caused by nitrogen or by sulfur, is to take samples, leaf samples, go to the lab, analyze. If the samples contain less nitrogen, then the deficiency is caused by nitrogen, and vice versa. This is very important because we once went to the fields, took samples. Before we took the samples, we were not able to actually know the element that is causing the deficiency symptoms unless you go to the lab, determine the two nutrients, whichever is smaller in quantity, then the deficiency symptom is caused by that particular nutrient. Thank you very much, sir.
Right.
Thank you, sir. Mr. President and Chairman of Council.
You are welcome.
Thank you very much for. I'm sorry to abandon
Standing for you. Yes.
Take over then.
Okay, sir. Thank you very much, sir.
Right.
Thanks a lot. Professor Amadu.
Yes, please.
Yes. You can go ahead, Professor Amadu.
Okay. I want to thank Trenchard Ibia for a very good presentation. In the area of the preliminaries, first step, and like we have always noted, if the first steps are done very well, then you can be sure of good results. First, the depth of sampling will very largely depend on the route you have in mind or your target crops. That's number one. If you have a shallow rooting crops like cassava that's still on the surface, you can as well limit your sampling depth to 20.
No.
Now, if you are sampling maybe adventitious and perhaps root crops, other ones that go deeper than 20, deeper than 20 or 15, you can as well get up to 30. Well, I don't know, getting to 40, you might be going out of the root zone or the so-called rhizosphere. My second observation is on the plant nutrition. And of course, people have made effort people have made effort to try to see if they can divulge soil fertility from plant nutrition. It's not usually possible because when you are looking for soil fertility, you must be looking at it with respect to a crop in mind or crops in mind. I so much appreciated the plant nutrient deficiency symptoms he did. I think perhaps some of our local crops, perhaps even if it's by using a camera in our phones, would have served a lot much better to show the younger ones deficiency symptoms as they manifest. But all the same, that was very good. Sometimes you find that even excess of phosphorus can give you a dark green color due to the anthocyanins that somebody will wonder what's going on. And then distinguishing between nitrogen deficiency, which expresses itself as mild, as distinct from those ones that are expressed by minor elements, trace elements, manganese, and perhaps the intravenous chlorosid from the zinc. That's a wonderful work he has done. But again, like we used to express here, soil fertility must go with plant nutrition in mind. I want to thank Trenchard Ibia for a very good work and the society for giving us all opportunity to refresh our minds and for the younger ones to discover new areas perhaps they have been ignoring. Finally, sir, please, I'm sorry. Critical levels are very important. If you go beyond if you have result beyond this, you are having excessive luxury consumption, sometimes even getting to toxic. And between regions, especially the agroecological zones in the north of this country, and perhaps in the south, and the use of strengths, BRE1 for the south, and BRE2 for the north, depending on concentration of the hydrofluoric acid and hydrochloric acids. Thank you, Trenchard Ibe, and thank you, Chairman of the Society, as well as Emeritus President of the Society, Raji. Thank you all for this wonderful job.
Thank you, Prof. Next question, Dr. Sam Meseli, please.
Thank you very much, Prof. First, I would like to appreciate the lecturer, that's Professor Trenchard Ibia, for the very good and comprehensive lecture he has given us today, at least to refresh our mind on the issue of soil testing, soil fertility, and fertilizer recommendations. Mine is more of a comment and a little contribution that soil fertility assessment should most of the time not completely focused on fertilizer application, because there have been cases where we evaluate the soil and you find out that in the end that the nutrients are sufficient, even to some even going above the critical level, even up to the level of toxicity, and you still realize that the crops are not doing well, and it is not because they are diseased, but just because some of these nutrients are already at the level of toxicity, like a case of boron. If boron is high and you also have the electrical conductivity to be high, so you find out that irrespective of the other nutrients, the crops will not do well. what is called the reverse osmosis tends to occur. So it's also good that the aspect of this nutrient toxicity, like Prof noted, is also very important. And like Professor Amadu also said, that the issue of fertilizer application, I mean, this issue of soil fertility should be highly related to a crop, because we find out that the soil that is fertile for a particular crop may not be fertile for another crop. So these are very important issues that we raised today. So that's just my comment, and to also appreciate NISS for this important opportunity to learn and refresh our mind. Thank you very much.
Libya.
Thank you very much. The comments, the observations, and the appreciations are very well taken. Professor Nwaka, methods of soil sampling, Asadu, methods of soil sampling, Professor Amalu, methods of soil sampling. So far, for fertility, we have been restricting ourselves to 30 centimeters. Like Professor Amalu said, it should depend now on the crop. If you are going to do soil fertility evaluation to classify a land type or classify an area and draw maps, you can go up to 45 centimeters, because it is possible that all kinds of crops can be grown thereafter. But I want to say generally that at the instance of our action group, action committee, NISS action, NISS 5-year action plan.
Working
groups, sir. We are looking at harmonizing all this. We have requested soil surveyors to give us details of how they sample soils in Nigeria. Okay. We have listed those in fertility to give us details of how they sample soils in Nigeria, in their area, their experience. So I want to believe that when these are put in place within a short period, we shall have standards that conform with international standards. But for now, like Mark observed, people just sample. A surveyor may write an article on fertility after doing soil survey, and it is published. A fertilitist may write an article with a surveyor, and so on and so forth. But I think we shall come out with standards that we say this is Nigerian standard across all the zones.
Okay. I will look at it again.
On the issue of I think Professor Gaba Umar has made a good suggestion. A lot of these symptoms look alike, and it has suggested that if you have symptoms similar to nitrogen or sulfur, bring it to the lab and look at the one that is more that is more or that is less. You can make inference from there. All what this points out to is that we have a lot of work to do. We have a lot of work to do, particularly minute details. We have not been taking care of minute details. And therefore, our laboratories must be furnished to take care of minute details. We have done so much in characterization. I think we should start looking at basic minimum of what does this really stand for. Professor Oche mentioned using local crops. Very good. We can only do that if we have capacity to first of all change our research focus. Plant nutrition cannot be separated from soil fertility. It is impossible. But that does not mean than somebody who is interested in nutrition alone, because people in chemistry and people in botany, some of them do research in plant nutrition and they publish.
Mm-hmm.
So we soil scientists must have absolute control of the one that concerns us and do it well. Critical limits and critical values again. Our harmonization document will bring out some of these things, and we may also have to start doing specific researches to confirm what we already know. Dr. Mesali made a good suggestion. We keep on looking at all these things as we progress. But let me say again that within the short period NISS has existed, a lot are being exposed to us. A lot of encouragement is coming in, and a lot of challenges are being open to us, and a lot of opportunities. So let us take part actively in all this. Thank you.
Thank you very much, Mr. Guest Speaker. I cannot see any other hand that has been raised, but on the chat platform, there is a question from Ogochukwu Onamdi asking if they can get a soft copy of this lecture.
Yes.
And I think the registrar and the guest lecturer will see to that. There are several other commendations.
It has been circulated.
Oh, it has been circulated. Thank you very much, sir.
It's been circulated.
They said the soft copy has been circulated. That answered the question. There are several comments on the chat group commending the guest lecturer and NISS for this program. So I can't see any other hand. Can we therefore like to appreciate the guest lecturer by putting our hands together for a wonderful lecture that has been also well delivered? Please, let's put our hands together for our guest.
Chairman, let us show the faces of everybody.
Thank you. Let's put our hands together.
Thank you.
Thank you very much. Thank you very much.
Thank you very much.
Thank you. Thank you.
Well,
Thank you very much, my coordinator.
Thank you.
Thank you very much. We appreciate you.
Thank everybody for listening.
Thank you, South coordinator.
Thank you. Thank you.
The next
Thank you very much.
Well done. Thank you. I appreciate it.
Thank you, sir. Thank you.
The next item on the agenda, please. I think it's a vote of thanks by the newly minted president of Soil Science Society of Nigeria. Okay. Vote of thanks by the newly minted president of Soil Science. Can we have him, please, if it's on the platform?
Okay, go.
And then the cancer rates are high, kidney failures are high, liver failures are high, congenital malformations are high. And we don't take care of that. So I just stand there and keep on looking up the bodies and keep on telling us
Okay.
Who is that?
Are you hearing me?
Oh no, we are maybe.
Are you hearing me, sir?
Hello, sir.
Are you hearing me?
And what we are eating is
Are you hearing me?
Yes, we are hearing you. Go ahead.
Okay, sir.
produce the things that we can eat. Everybody
it doesn't matter. The mother not inside there.
Go ahead, Prof.
Okay. My name is Professor Yinka Okubeno Dipelu, Vice President of representing the President of Soil Science Society of Nigeria, Professor Jubre. Yes, we appreciate the guest lecturer in person of Professor Trechard Okon Ibia, who is the South-South coordinator, the NISS South-South coordinator. We appreciate you, sir. You actually dealt with the topic, and it was so glamorous in a glamorous way. We appreciate you. Thank you very much, sir. On behalf of NISS, I want to appreciate the president of NISS, the chairman of the governing council, Professor A.O. Bukole, the RSS, FSN. We appreciate you for the time being there to be part of this. We appreciate you, sir. Thank you,
Thank
sir.
you very much.
We want to appreciate the Vice President of NISS Governing Council in person of Professor G.I.C. Nwaka. Thank you, sir. You are presently now the chairman of NISS Education Committee. We appreciate you, sir, for coming.
Thank you very much for appreciation.
Being part of April's occasion. We appreciate you, sir. I want to appreciate no other person again. The person of the registrar, the Chief Executive Officer of NISS, in person of Professor V.O. Chudi. We appreciate you for all the effort put in place to make this memorable day. We appreciate you. We appreciate each and everyone. I mean the participants, Raji. We thank you. We thank each and everyone, all professors present here today. We appreciate you for coming. We thank you, Lord, for sparing your time to be part of it. And we thank each and every member of NISS for their participation, taking time to listen. We pray that God, in His infinite mercy, will continue to prosper and make NISS to be progressive. Make NISS to continue to impact as regards soil cases in Nigeria and all over the world. Thank you and God bless.
Thank you very much, the President of Soil Science Society of Nigeria, ably represented by the Vice President. The President has thanked everyone from the President of the Council to the Vice President, the Registrar, all professors and distinguished registered soil scientists. Let me, on behalf of the ICT crew, apologize for the little hitch we had. Part of the technology, and I believe that will continue to get better.
Next time, let them show us pictures of everybody.
Thank you. So the Professor Amali is asking for pictures. I don't know.
Prof, I learned that you were a delegate.
Maybe
I learned that you were a delegate.
I was there, but I collected only 10 million.
So we thank the President. Then we give God to the first series. Thank you very much, Mr. President. And thank you, Registrar.
Come and kidnap me.
He wanted to contest for Anambra governorship.
Yes, so.
Don't turn this forum to breaks.
Okay. Thank you.
Bye.
Bye.
God bless you.
Well done.
Thank you.
Thank you very much.
Bye-bye.
Bye.
Bye. Bye, everybody.
Bye-bye.
Bye.
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 Bye. Bye.
Thank you.
Our network was good. The network, the most important. And thank God.
Charles Asado, I salute you.
Well done.
Thank you, everyone. This lecture, you should have more of it. Thank you.
Thank you.
Thank you very much.
Thank you all.
Thank you.